All right, this is the nervous tissue chapter. The nervous system. There's actually two major systems for the body to communicate. One is the nervous system, and one is the endocrine system. The endocrine system uses hormones dumped in the blood, and it's significantly slower relative to the nervous system. Nervous system communicates by neurons and is significantly faster, much faster. The nervous system can be divided into the central nervous system, where you have the brain and the spinal cord, and then the peripheral nervous system, or the PNS, CNS, and PNS, where you have neurons in the arms, the legs, the torso, and so on. Picking up information is done by the sensory receptors. We have somatic sensory receptors, visceral sensory receptors, and special sensory receptors. The somatic, like position, provide position, touch, pressure, temperature, pain, and so on. The visceral sensory receptors monitor internal organs, and finally, the sensory receptors provide sensations of smell, taste, vision, balance, and so on. If we want to know the actual names of the receptors, we have interoreceptors that monitor digestive, respiratory, cardiovascular processes or sensations. We have proprioceptors, which monitor the position of the movement or where we are in space, our arms, our legs. We know if our hands are open or closed, or if our legs are extended or flexed without opening our eyes or looking, because of these proprioceptors. And we also have exteroreceptors, which provide information about the external environment in the form of touch and temperature, pressure, and so on. You guys remember, for example, the Pacinian corpuscles, or the Meissner's corpuscles. Those are examples of exteroreceptors. This entire direction from the PNS to the CNS, we talk about the sensory division. Sensory moving towards the CNS, and information moving towards the PNS from the CNS back out to the PNS we call motor neurons, the motor division. And it can be divided into the somatic nervous system and the autonomic nervous system. The somatic nervous system innervates skeletal muscle, whereas the autonomic nervous system innervates smooth muscle, lining organs, for example. Innervates cardiac muscle, glands, adipose. The typical neuron has typical regions. We have dendrites that receive information, or even the cell body. The dendrites, here we have 5. 1, 2, 3, 4, 5. We got the cell body, and in the cell body we have nucleus. And these, neurons are in many ways are no different than any other cell in our body. They have everything. They have mitochondria. They have Golgi. They have endoplasmic reticulum. Just like the typical cell that we generally study in general biology. We have the cell. So we have dendrites. We have the cell body. We have this special segment right here called the Axon Hillock. Axon Hillock is the first segment of the axon, sort of right in this area here. That's the Axon Hillock. And we have the axon, and this is obviously to fit this all in one screen. A neuron can be 2-3 feet long. That's long. You know, you think of a cell as being, you can't see a cell macroscopically. You know, neurons can get very several feet long. This axon could be extremely, you know, this is obviously not to scale. And then finally we have the terminal buttons, or the axon terminal right here. The connection between the first cell and the second cell is called the synapse. And the second cell may not be a neuron, it could be a muscle, you know, where this would be the neuromuscular junction. So we can call this a presynaptic cell and a postsynaptic cell, or a presynaptic neuron and a postsynaptic neuron. But of course, the second cell is not necessarily a neuron. It could be something different. And then at the end, inside that the Golgi and the endoplasmic reticulum make, that the cell make, you'll have chemicals in here. Different types of neurotransmitters like epinephrine, norepinephrine, acetylcholine, and they're stored right here in these ends in these little bags called vesicles, ready to be released out of the presynaptic cell and into the synapse, which is the little gap between the two cells. There are 4 anatomical classes of neurons. We have anaxonic, or no axon cells, and you just have to know just a couple of things about them. These are found in the brain. They're also found in special sense organs. Other than the axon, the cell looks pretty much the same. We have dendrites here, we got the cell body, we got the nucleus right there. We also have bipolar axon right here. Here's a dendritic process. Here's a dendrite here. Here we have an axon, and I forgot to mention in the first in this first diagram here that there is always a direction. Orientation-wise, there's always a direction. Since information is picked up in the dendrite, information moves, in this case, from left to right. Presynaptic cell, postsynaptic cell. And so all you have to do is just figure out, look and see where are the dendrites, where's the axon terminal, and then you automatically know which direction. It's always only one direction with these cells. So here, we look for the dendrites, we look for the axon terminal, and we know automatically here, this would go from top to bottom. The information would go this direction. So we have bipolar. And these are rare. The little piece of information that I'm going to require you to know is that these are rare, but they are found in special sense organs. We also have unipolar axons. Sorry, unipolar neurons. And most of the sensory neurons of the PNS that are going up towards the CNS, most of the neurons in the PNS, the sensory neurons in the PNS, are unipolar. And finally, we have multipolar. These are this is the typical neuron that we see depicted in textbooks and so on. This is the typical one. This actually happens to be a multipolar neuron, and this is by far the most common, what we typically find in the CNS. So we have CNS. We have, these are generally found in the CNS. These are generally found in the PNS. These are rare, but they are found in special sense organs, and these ones are found in the brain and in special sense organs. Now, where do these names come from? We have anaxonic, oh yeah, that means no axon, but bipolar. Well, if you take a look at the cell body, we have 2 poles coming off. They're not 2 axons, but they are 2 poles. One of them just happens to be a dendrite. One of them happens to be an axon. We have unipolar, but I don't see one pole. Actually, if you look closely, the cell coming off of the cell body is just one single pole. Therefore, this entire thing is actually an axon here. Unipolar, because the cell body is not in line with the axon, but is hanging off. Here, the cell body is in line. Here, it's actually hanging off. Unipolar, the cell body hangs off. And multipolar, well, once again, you take a look at the cell body. And look, we have 1, 2, 3, 4, 5 dendrites, and we have a 6th axon. So multipoles coming off of off of the cell body, and that's where those names come from. Multipolar, multiple poles coming off of the cell body. Bipolar, 2 poles coming off of the cell body. Unipolar, 1 pole coming off of the cell body, and then this one is anaxonic. We have 3 major functional classes of neurons. We have sensory neurons coming up. We have motor neurons leaving the CNS into the PNS, but we also have interneurons, which often bridge the sensory neuron with the motor neuron. We find this interneurons in the CNS as well. So we have these sensory moving from the PNS to the CNS, and then interneurons connecting the two, and then motor neurons leaving the CNS and into the PNS. We have 4 CNS neuroglia. We have 4 CNS neuroglia. We have ependymal cells, and these form epithelia, an epithelial lining. The function of these, let me just zoom in here. The function of these is that they make and circulate, they produce and circulate and monitor CSF, cerebrospinal fluid. Ependymal cells. Number 2, we have microglia. And microglia are embryologically related to monocytes and macrophages, the white blood cells in our blood. They actually are mobile. They float around. These are the only ones that actually float around the brain. They go around seeking and destroying. They go around looking for pathogens, cellular debris, waste products, anything that poses a threat. These are basically the white blood cells of the CNS, and they are actually embryologically related to monocytes. And how do they seek and destroy? They engulf. They by phagocytosis, they engulf the pathogen and destroy. Those are microglia, mobile neurons. Mobile neuroglia. We also have astrocytes. The function of these is that they maintain the blood-brain barrier, isolating the CNS from the chemicals and hormones in the blood, and they also provide structural support of the neurons. And finally, we have oligodendrocytes, and these make myelin. You see this jacket here, this wrapping sheath of myelin? This is actually made out of omega-9. We know we're familiar with omega-3 and omega-6. This is the brain. 90% of the brain is actually made out of omega-9. We can convert it. We can just consume whatever, and then we have enzymes that convert it into omega-9. And wherever you find this fat, this omega-9, you have white matter. We hear of this white matter and gray matter. The gray matter is just void of this myelin, these myelin sheets. And where you have white matter, it's white because it's filled with these jackets of myelin. So it's the oligodendrocytes that produce this myelin. But remember, we're talking only in the CNS. These 4 cells are in the CNS. One more time, we have ependymal cells making and producing and maintaining cerebrospinal fluid. We have astrocytes, we have microglia that seek and destroy cellular debris and pathogens by phagocytosis. They float around, move around in the brain. We have astrocytes that maintain the blood-brain barrier and provide structural support for neurons. And finally, we have oligodendrocytes that produce myelin. And the function of these jackets then, these myelin sheets, is to allow for a faster transmission of information. It's called propagation. It's called propagation because fat insulates but doesn't conduct. The signal is forced to jump around this, and therefore with this jumping, the signal moves significantly faster. It's propagated. The signal is propagated faster. And there is your 4 CNS neuroglia. We also have 2 PNS neuroglia, peripheral nervous system. Now, so here's a neuron, and what do you know? We also have, it looks like, more myelin. Schwann cells. Now, Schwann cells in the PNS, not the CNS, Schwann cells in the PNS also make myelin, these myelin jackets. And here we have these nodes, and the signal will then jump. So typically in the PNS, we have much faster signal than a lot of the CNS. And then we also have satellite cells. And just as you would expect, satellite cells are found around the neurons and these regulate intracellular environment, much like the astrocytes. So we have 2 PNS neuroglia. We have Schwann cells, and we have satellite cells. If an axon just happens to be, if a neuron happens to be damaged, it is possible to repair a neuron. The distal end of the neuron will degenerate. Schwann cells then will start to wrap myelin and provide sort of a tunnel for the axon to find its way and make its way to the effector, to the muscle or whatever organ. Only if the Schwann cells can wrap and form a tunnel. If not, then you just lose that you lose that neuron. And if you cut enough, it might become permanent. You know, like in a C-section, you could lose some some sensation. You have some tingling, for example. So there is repair in neurons, but it's limited. They're not like epithelial cells where they're readily dividing and undergoing mitosis. These cells don't undergo mitosis. They're not constantly dividing like epithelial cells. But there is some repair, but it is limited. All right. Now, if we zoom in to the cell now, one particular cell, if we zoom in, we'll find that the inside of the cell is slightly negative and has a slightly negative charge, and the outside of the cell has a slightly positive charge. And then we have the plasma membrane here. So this would be the inside and this would be the outside. We have several potentials. We have resting membrane potential, we have graded potential, and we have an action potential. At rest, the resting membrane potential, the inside of the cell is slightly negative. You need to know the exact number, and that number at rest is negative 70 millivolts. That is due to the ions being inside, negative 70 millivolts. Inside the cell, we will find a whole bunch of ions. Mostly what we end up finding inside are potassium, 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 potassium. Inside the cell, at rest, again, at rest. So if you're drawing along here, put a whole bunch of potassium, mostly, I mean, there is some outside and there's some inside, but mostly inside. If we're just trying to depict what the cell looks like at rest. So I'm going to put a lot more potassiums inside. Okay. Outside the cell, we have a whole bunch of, what do you think? Sodium. So we have some sodium. We have some sodium inside as well, but mostly we find that outside. So if you're drawing along, both have a positive charge. They both have a positive charge. How can we remember that sodium is mostly outside and potassium mostly inside? Well, salt is bad for you, right? Salt, of course, is not really bad for you. Too much salt is bad for you, but that's how we can remember. Sodium outside the cell and potassium, bananas, right? Inside the cell. So at rest, you have to remember that potassium naturally will find a lot more potassium inside and sodium a lot more on the outside. Okay. Now, take a guess. You have to know about these guys right here. We have sodium leak channels. We have potassium leak channels. Well, just with our intuition now, if we have a whole bunch of sodium outside the cell, they will leak which direction? They will leak in just by diffusion, just by diffusion. If you have a whole bunch of stuff outside, they will naturally want to come in, and that's exactly what they do. Sodium will leak in. Not a whole bunch, but just a little bit. This is part of the regulation. Sodium leak channels. So you have to know that sodium leak channels leak sodium in. We also have potassium leak channels. And if most of the potassium sits inside the cell, then that means that potassium leak channels will leak potassium, which direction? Out. Now, if this is the only mechanism, then we're going to find all the potassium out and all the sodium in, and then that's the end of the story. What is keeping all the potassium, what is pulling all the potassium back in and all the sodium back out? The sodium-potassium pump. We got the sodium-potassium pump all along the membrane. In the dendrites, in the cell body, along the axon, we have the sodium-potassium pump. And again, once again, you have to know which direction the sodium-potassium pump pumps sodium and which direction it pumps potassium. It pumps sodium which way? We're using energy now, so it pumps sodium out, and it pumps potassium in the way we want it. So now we've just talked about, 4 different mechanisms. We have 2 leak channels, and we have the sodium-potassium pump. 4 different directions these things can go, or 4 different, in different instances. So you have to know, you have to keep those straight. All clear? All right. And it's also good to know that for every 3 that the sodium potassium pump using ATP, for every 3 sodiums that it pumps out, it pumps 2 potassiums in. But both have a positive charge. Both are positively charged. What's the resting membrane potential? -70. Okay. When information is taken in, when information is taken into the cell, when the cell senses information, it moves this direction and it takes in information by a mechanism called graded potential. You have this signal that is graded. In other words, it weakens as it travels towards the axon hillock. You see that? That one graded potential might be strong enough for the axon hillock to say, go, give the green light, and fire, and cause an action potential to go all the way down the axon, through the axon terminal, and to the next cell. If the axon hillock gives the green light, the signal will always move down the axon and it will always reach the next cell. That signal might be strong enough, and it also might not be strong enough. It might need, for example, a second signal coming in. So multiple dendrites might be what it needs for this axon hillock to give the green light. There's also another way that might increase the chances of this axon hillock firing. If the signals coming in are closer to the axon hillock, then that increases the chances. If they're further away, that decreases the chances. Does that make sense? At what voltage does the axon hillock need to be at in order to fire? It needs to be at this term called threshold. It needs to be at -60 millivolts on the inside of the membrane. inside the cell. We have three types of gated channels. We have chemically gated channels, we have voltage gated channels, and we have mechanically gated channels. The chemically gated channels, we find the chemically gated channels. Here's an example of one chemically gated channel right here. We find chemically gated channels in the dendrites and on the cell body. So I want you to know where we find them and how do they work. The chemically gated channels open when they bind specific ligands or specific chemicals, just like acetylcholine, for example, a neurotransmitter. Where would these come from? They would come from the cell from before, or from some other part of the body. These are also stored in the synaptic terminals, like we discussed before. These are generally found. So where are these generally found? These chemically gated channels. Where are they generally found? The dendrites, cell body, not the axon, but the dendrites, the cell body. What moves through them is the same stuff that moves through the voltage gated and the mechanically gated. This is the stuff that moves through, but how they are activated is different. So how are they activated? By ligands, by chemicals attaching to them. Then they can open, if they're the right shape, then they may open and then let the same stuff move through. Sodium, potassium. We also have voltage-gated channels. We also have voltage-gated. Voltage-gated channels we find along the axon. We find along the axon. Voltage-gated channels allow the same things to move through them, sodium and potassium, but they are found in different, but they are not found in the dendrites and the axon and the cell body, but in the axon. And then we also have mechanically gated channels. We're gonna go into detail now with the voltage gated channels. That's gonna be basically the rest of the talk. Is gonna be how these guys work. So we also have mechanically gated channels, and these are found way back here again in the dendrites and the cell body, and they open in response to physical distortion, mechanically, physical distortion, and these would be examples of touch and pressure and vibration, just like Meissner's corpuscle, Pacinian corpuscle. And so if we wanted to draw an example of this, they would look something like this. And when the membrane is physically distorted, they would open up and allow the same ions to move through, sodium and potassium. So these are mechanically gated channels. We have chemically gated channels, and we have voltage gated channels. All right, quick question for you. If we find graded potentials occurring in the dendrites and the cell body, and we have action potentials occurring along the axon, which of these gated channels allow for graded potentials or local potentials. Which ones? Chemically gated and mechanically gated. These allow for graded potentials because graded potentials only occur in the cell body and the dendrites. Graded potentials or local potentials. Here, the voltage gated channels, since these are found along the axon, and since the axon allows for action potentials, that's the voltage gated channels that cause or allow for action potentials to occur. So now we know what these three do. We know what goes through them. We know how they're activated in general, and then we know where they are and where they are found. Now we have now in the voltage gated channels. The voltage gated channels, they're not channels now, but they're gated, meaning they have doors. Here's a door, here's a door, here's a door, another door. In other words, they open and close, just like these would open and close as well, but these don't really have doors. They just move apart and away from each other. These actually have physical doors. The potassium channels, the potassium gated channels, have one door, but the sodium gated channels actually have two. They have an activation gate and an inactivation gate. I've made a little graph here to show the voltage, to show the voltage of the inside of the cell at the various at various times. Here on the y-axis we have the voltage. These are millivolts here, the transmembrane potential in millivolts. And along the x-axis here, we have time. What did we say the voltage was inside the membrane at rest? Negative 70. So here it is. Now, we remember when we said what occurs during graded potentials? There might not be a strong enough signal that reaches the axon hillock. And here's an example. Here's where the membrane goes positively, but then goes back. It's not strong enough, and it goes back down to rest. How did it go back down to negative 70? Which of those channels that we've talked about so far? Which one? The leak channels? The sodium potassium pump, pumping everything back like it was at the beginning. Well, let's just pretend, along with this graph, that down here is the inside of the axon, and out here on top of this graph is extracellular fluid. So I sort of used this graph to sort of depict the one neuron. Where is sodium occurring? Outside. Where is sodium at rest? Inside. Let's take a look at the gates. Now, where are we here? Where in the cell are we? Where exactly are we? Not the cell body. We're actually already on the axon, because the voltage gated channels are found on the axon. You guys see that? Okay, so at rest, this is what the two gated channels look like. They are both closed. This is the activation gate outside. The outside one is the activation gate, and the inside gate is the inactivation gate. This one is the inactivation activation gate. And this one is the activation gate. Right here. Notice the inactivation gate is open and the activation gate is closed. There only needs to be one that's closed in order for things not to flow through. Well, the inactivation gate at rest generally is. Now, we have these channels. We have these gated channels all along the axon. That we'll find them. We find them all along. You don't just have one or two. There's many, 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 many. And so when a signal is propagated along the dendrite and the cell body, and the axon hillock gives the green light. The question is, which of these channels which of these gates will open and allow for the signal to be propagated? Anybody know? It's always the same order. In muscle, in nerve, no matter what, it's always the same order. Which one? The sodium. The sodium activation gate opens and sodium rushes in first. Sodium rushes in first. Sodium rushes in. And of course, you have to remember which direction. And here we go. Here is what it looks like when it's open and sodium begins to rush in. Now, why does the graph begin to go up? Because sodium is a positive charge, and so it's going up. So the graph goes up. Now, actually, this is not 100% accurate here, because actually -60 was threshold. So actually, these gates should be up here, and then we should have mechanically gated channels here and chemically gated channels down here, right? Threshold. If threshold is reached, -60, it's still in the negative. It's still negative, but it's much more positive than -7 than rest. If that occurs, then the action potential will always go, and it's called. And this action potential has this principle called this all or none principle. Either it fires or it doesn't. If it fires, the signal will move all the way down the axon to the axon terminal, and the signal will always reach the next cell, always. The graded potentials can begin but they die off. They're graded. All right. Sodium rushes in, and this causes the cell to become more and more and more positive. And so the cell moves in the positive direction. It's still, even at this point, the cell is still, look at where the millivolts is here, it's still negative, but it's moving in the positive direction. The cell will continue to move up, up, up until the third number that is important to note is +30. +30. So we have -70, which is rest. We have -60, which is called threshold. Then we have +30 is the next number of interest. What happens at a voltage of +30? What occurs? Yes. When the cell is way down here and begins to move up, that is called depolarization. When it begins to become negative again, that's called repolarization. But what exactly is occurring? Where is the sodium at Once the cell reaches here, where is sodium? It's inside. We just got done saying that it was rushing in, rushing in. Where at positive 30 or positive 20, where is potassium? At positive 20, for example, or positive 30, where is potassium? It's also inside. We haven't talked about potassium yet. Potassium at rest was inside. So this entire time that the cell was depolarizing, the potassium wasn't moving. Now, which direction does potassium then move? It moves out. And because potassium is what charge? Positive, and it moves out, the cell becomes more, it moves in the negative direction, which is exactly why this graph moves down, 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 because now potassium is moving out. Yes, sodium is in, but potassium is moving out. And actually what happens, now here's threshold, and here's rest. Notice how far down the graph actually goes. It goes even further down to, past negative 70, maybe even negative 80. Why does that occur? That occurs because it takes a little bit of time for these potassium gates to shut. It takes some time, and so the cell actually hyperpolarizes, and so this region here, this spot is called hyperpolarization. Hyperpolarization. It actually becomes more polar than it was at rest. Now, by what mechanism does this graph go back to 70? Or let me ask you, where is sodium now? Inside. Where is potassium? Outside, right? Sodium was outside. It rushed in. At positive 30, potassium now rushes out, and down here, that's exactly what we talked about. Well, now potassium is out and sodium is in. What allows for the cell to go back to how it was before? It's the sodium-potassium pump that pumps everything back in the original position, the original way. Pumping sodium back out and pumping potassium back in. And the ratio 3 to 2, once again. Now there's a couple more terms that I want you to be familiar with. If we draw a line here, and another one right around here, we have a couple of regions. We have what we call absolute. Right here we call, let's see here, let's draw this, let's write this in green. We call this absolute refractory period, and we call this region here relative. Refractory period. What does that mean? It means, and remember, this whole process that's occurring, this entire thing occurs at every tiny little segment all along the axon. This whole thing doesn't occur just one time along the axon. No, it occurs hundreds of thousands of times, because we have sodium and potassium all along the So this whole thing will happen again and again and again and again, over and 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 over. Each little tiny segment of the axon goes into depolarizes and repolarizes and depolarizes and repolarizes and goes into absolute and relative and absolute and relative over and over and over and over and over and over and over hundreds of times in a single action potential. That makes sense? What does absolute refractory period mean? It means that in that little segment of the axon, in that little segment, the cell can absolutely not fire again. That's what that means. It absolutely cannot start over. However, do you notice in this line, this border here, we actually aren't at threshold or even at rest. We're at the graph hasn't yet dropped. I didn't draw that line over here. Relative refractory period means that in that segment of the axon can actually fire once again. However, it needs a greater than normal stimulus to fire. It didn't go all the way back down to resting yet, or even to threshold, but it's close enough that it can fire again. It just needs a greater than normal stimulus. Does that make sense? So we have depolarization until the top of the graph. We have repolarization until threshold or rest. We have hyperpolarization that because the voltage of the inside the cell passes -70. It begins to move, the cell begins to move, and that segment of the axon begins to move in the positive direction because sodium channels open first. And then it drops because potassium channels open. Now, let's take a look at this activation and inactivation gate. Which one opens first? The activation gate opens and allows sodium to go in. Notice at the top here, now sodium is inside completely, and the inactivation gate now comes into play. The inactivation gate closes, and just like the word says, it inactivates the movement of sodium. The inactivation gate was open that whole time, and then it closes. It closes, while the activation gate is still open. Then, once everything goes back to normal, the inactivation gate can then open back up, and the activation gate can close, just like at the beginning of when it was at rest. So, at rest, the activation gate is closed, the inactivation gate is open, but when at positive 30, in order to stop the flow, that's the function of the inactivation gate. It inactivates the flow of sodium in, and then once the sodium-potassium pumps have pumped everything back to normal, these gates can go back to their original state. That makes sense so far? All right, we have a couple of terms. We have continuous propagation, and we have saltatory propagation. Continuous propagation occurs along unmyelinated axons. So this generally happens in the CNS, unmyelinated, because most of the neurons in the PNS are myelinated. And the speed is about 2.2 miles an hour, or 1 meter per second, which is pretty fast. That's like walking speed. However, in saltatory, not continuous now, but saltatory propagation, the speed is significantly faster at 447 miles an hour, or 200 times as fast, 200 meters per second. And that's saltatory, coming from the word leaping. why do we have leaping and why do we not have leaping? Well, in the axon, in axons where there is no myelin, there is no fat jacket wrapping, the gates are very close to one another all along the axon. So what occurs here, let's take a look here now. Now we're going to see that we're going to look at the same process except without the gates now. We're just going to see the movement of ions. Check this out. Sodium rushes in, and the charge and the voltage begins to become more and more positive. Here's rest, -70. Sodium rushes in, and because sodium rushes in, it's attracted to the negatively charged inside due to the proteins and so on. And so it starts to spread. There's a local current in this direction. And because sodium is being is rushing in, it's actually pulling sodium from over here and dumping it, and it's being pulled this direction because these charges are attracted. And what does that do? It causes the membrane to become more positive from -70 to -60. And -60 is actually threshold at this exact point. Therefore, more sodium gates will open up and cause more sodium to rush in. And the same thing will occur over and over and over. It'll continuously move in that direction. And that's why at these points we have absolute refractory period, because the gates are still open. You have that, you can never go in the backwards direction. Now, if we look at saltatory propagation, these are Schwann cells we're looking at here. This is the myelin that we're looking at here. The same exact, there is absolutely no difference between saltatory and continuous propagation other than that the ions move faster because there is fat in the way. And these things are in the way here, these internodes. Here's a node, here's a node, here's a node, node. And so these sodium ions, because there is this internode in the way, this myelin sheath, the ions move further faster in order to get to the same location, because they are attracted again to the negatively charged inside. And therefore it occurs, and therefore the signal travels so much faster. And if we look at a comparison between the two, if we take a look at a cell that is not myelinated in t equals 10 milliseconds here, the signal will have traveled, say, 1/3 of the distance, compared to in the exact same time if a cell is myelinated. Look at how much further the signal has traveled, has been propagated. We're almost to the terminal buttons over on the side. And that's the difference between unmyelinated and myelinated. What occurs here? Continuous propagation. What occurs here? Saltatory propagation. That really is the meat and potatoes of this chapter. Now, we also have one more ion that we talk about, which is calcium. Calcium is waiting at the terminal buttons. We talked about sodium, we talked about potassium. Calcium is waiting at the terminal buttons, and it's the calcium that allows for that rushes in when the signal has reached the terminal button, it's calcium that allows for the vesicles to merge with the membrane at the end here and release the contents, the neurotransmitter. The neurotransmitter then fuses with these chemically gated channels on the postsynaptic cell, and that is excuse me, and that is actually where we began the physiology of in the muscle chapter. That was action potential number 1 that sweeps down the sarcolemma. You remember that? And so now that sort of completes the nervous system with the muscular system physiology. So we have sodium. You need to know where does it sit at rest. Potassium, where does it sit at rest? Calcium, where does it sit at rest? Which direction do they move? Which channels allow them to move in, out, and so on. We also have excitatory postsynaptic potentials. We have inhibitory postsynaptic potentials, and these are graded potentials in the postsynaptic cells in response to the neurotransmitter. In other words, some of these signals cause the postsynaptic cell to fire. Some of these potentials cause the postsynaptic cell to not fire, to make sure that it doesn't fire. In excitatory postsynaptic potentials, the graded depolarization caused by the neurotransmitter arrival. Here is on a graph, these potentials cause the cell, the postsynaptic cell, to become close, to go in the positive direction, closer to threshold, whereas inhibitory postsynaptic potentials cause the postsynaptic cell to actually go in the negative direction to ensure that the cell doesn't fire. You see that? So that way it causes inhibition. In other words, you need a larger than usual stimulus to reach threshold now with these IPSPs. Okay. Here's what the end of a cell actually, or the beginning of a cell actually looks like. If this is the cell body here, and these blue ends are the dendrites, look at how many synapses are, and how many axon terminal buttons there are coming in. This is more like what it looks like, not just, one cell to one cell. This is more what happens. We have a couple of terms. We have summation, and summation is the integration of graded potentials. And so here again, we have several of these all in play. A lot of times we have many of these in play at the same time, talking to a postsynaptic cell. Many different neurons talking to a single postsynaptic cell. Well, one single EPSP or a single IPSP has a very small effect, maybe roughly a half of a millivolt, and it takes about 10 millivolts in order for it to fire. And finally, we have two types of summation. We have temporal summation and we have spatial summation. Temporal summation is a single synapse stimulated repeatedly. Firing, 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 firing. We see here we have a single synapse, one terminal button. Here is the postsynaptic cell here. Here's the axon hillock here. And it's one neuron firing, 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 you know, just like that. And we also have spacious, so that's temporal summation, and we also have spatial summation. And this involves multiple synapses activated simultaneously. And of course, the degree of depolarization depends on the number of synapses and also the distance from the initial segment or from the axon hillock. So here's an example of spatial summation where you have not necessarily one firing multiple, but two different ones firing once, but you also it's also the signal is summed or added up, increasing the chance for this signal to go down to cause an action potential. And that concludes the nervous tissue chapter.